Up the down staircase, Diencephalon. The diencephalon is the part of the brain between the midbrain and the cerebrum. It consists of the dorsal part, including the thalamus, epithalamus, and the metathalamus, and the ventral part, including hypothalamus and subthalamus. The thalamus consists mainly of gray matter, which is divided into anterior, medial, and lateral parts, each in turn consisting of several nuclei. The diffuse distribution of large and small neurons was demonstrated in almost all nuclei. However, several have distinct magnocellular and parvocellular parts. For example, the medial dorsal nucleus, which has medial magnocellular and lateral parvocellular part. The metathalamus consists of the medial and lateral geniculate bodies. The lateral geniculate body consists of dorsal and ventral nuclei. The dorsal lateral geniculate nucleus consists of 6 layers that are divided into 4 dorsal parvocellular and 2 ventral magnocellular layers. Each layer receives inputs of one hemiretina either from the ipsilateral or contralateral eye, covering the contralateral visual field. For example, the right dorsal lateral geniculate nucleus receives inputs from the right hemiretina of both eyes that correspond to the left visual field. Layers 1, 4, and 6 receive input from the contralateral eye, and layers 2, 3, and 5 receive input from the ipsilateral eye. All layers are retinotopically aligned, that is, a line penetrating perpendicularly to the layers passes through receptive fields covering the same spot of the visual field. Ventral to each parvocellular and magnocellular layer, cornea cellular layers consisting of very small neurons are located. In Greek, cornea means dust. Some corneocellular neurons process short wavelength visual information, some of them process low acuity images. The medial geniculate body consists of ventral or principal, dorsal, and medial nuclei. The ventral nucleus of the medial geniculate body is arranged in laminae according to the tonotopic organization, as the central nucleus of the inferior colliculus. The ventral nucleus receives input from the central nucleus of the inferior colliculus and the auditory cortex. The ventral nucleus has several specific features. Firstly, it possesses many GABAergic Golgi type II cells whose axons do not leave the metathalamus, being intrinsic inhibitory neurons. Secondly, it has synaptic glomeruli in which processes from Golgi type II neurons, besides axons from the inferior colliculus, are presynaptic to projection neurons, these neurons being surrounded by a glial sheath that isolates them from neighboring influences. Nerve fibers arising in the ventral nucleus form the auditory radiation and run to the brain cortex. The epithalamus includes pineal body, lateral and medial habenular nuclei, stria medullaris of thalamus, habenular commissure, and posterior commissure. The pineal body or pineal gland is covered by a tissue capsule whose layers penetrate the organ, branch and divide the parenchyma into lobes. The parenchyma of the pineal gland contains pinealocytes, astrocytes, and microgliocytes. The pinealocytes are larger than astrocytes, have a polygonal shape, light nucleus with large nucleoli. Near the capillaries, the long processes form club-shaped extensions with secretory granules. Pinealocytes synthesize melatonin using enzymes that are sensitive to diurnal changes of light. Level of melatonin synthesis increases in the absence of photic stimulation in the night. Daylight decreases the enzymatic activity and melatonin production is decreased. Besides the melatonin, pinealocytes also produce serotonin, norepinephrine, and thyrotropin-releasing hormone. All secretory products are released into the blood circulation and the cerebrospinal fluid. Indirect photic stimulation of pinealocytes occurs in the following way. Intrinsically photosensitive retinal ganglion cells project to the suprachiasmatic nucleus of the hypothalamus. In the darkness, the nucleus stimulates neurons of the spinal cord intermediolateral nucleus through pathways including paraventricular and brainstem reticular nuclei. The preganglionic sympathetic neurons of this nucleus project to the superior cervical ganglion, whose postganglionic neurons innervate the pineal gland, which produces melatonin. On the contrary, when the light signal is positive, the suprachiasmatic nucleus inhibits postsynaptic neurons and melatonin is not synthesized. Thus, the production of melatonin by pinealocytes corresponds to the circadian rhythm of the body that regulates the sleep-wake cycle, influences hormone release, eating habits, digestion, body temperature, and some other functions. The circadian rhythm is generated by a biological clock, that is, an innate timing device consisting of specific molecules that interact with cells in all tissues and organs of the body. In mammals, the suprachiasmatic nucleus is a major pacemaker or master clock for circadian rhythm phenomena. It coordinates all the biological clocks in a living organism, keeping the clocks in synchrony. But internal circadian timer of the suprachiasmatic nucleus is longer than 24 hours. It is synchronized to 24-hour circadian rhythm by various factors, among which melatonin is a major chemical message. Melatonin membrane receptors were identified on the neurons of the suprachiasmatic nucleus, and feedback information of melatonin produced cyclically by the pineal gland helps the suprachiasmatic nucleus to properly interpret the natural photoperiodic changes and to adjust its circadian rhythm to 24 hours. Besides the described action on circadian rhythm, melatonin has various functions such as antioxidant and anti-aging properties, immunomodulatory actions, differentiation, proliferation, regulatory effects, regulation of carbohydrate metabolism and glucose homeostasis, anti-tumoral activity. Pineal-derived melatonin is involved in the endocrine modulation of human sexual maturation. Nocturnal secretion of melatonin exerts an inhibitory role on the hypothalamus and pubertal maturation. It has been reported that before puberty, even if they progressively decrease, melatonin concentrations are too elevated to allow hypothalamic activation. However, at 9 or 10 years of age, the decline of serum melatonin gives positive signal for hypothalamic activation and the onset of pubertal changes. Unfortunately, the pineal gland has the highest calcification rate among all organs and tissues of the human body. Calcification jeopardizes synthetic activity of the pineal gland. It is associated with aging and may lead to a variety of neuronal pathologies. In addition to synthetic activity, the pineal gland also participates in cerebrospinal fluid production and recycling. The blood filtration rate of this gland is comparable to that of the kidney. In this regard, the pineal gland belongs to circumventricular organs. The circumventricular organs are structures located around the third and the fourth ventricles and characterized by a lack of blood-brain barrier. They facilitate the transport of peptides, hormones, and other bioactive substances between CNS and peripheral blood. Circumventricular organs are divided into sensory, that sense plasma molecules and pass that information to other regions of the brain, and secretory, that secrete hormones and glycoproteins into the blood. The subfornical organ, the vascular organ of the lamina terminalis, and the area postrema are sensory, whereas the pineal gland, median eminence, neurohypophysis, and the subcommissural organ are secretory circumventricular organs. The gray matter of the hypothalamus consists of numerous hypothalamic nuclei. Due to their various connections with different parts of the CNS, the hypothalamus ensures many important functions, including control of autonomic and endocrine activities, neurosecretion, regulation of food and water intake, control of emotional behavior and response to stress, regulation of sexual behavior and reproduction, memory preservation, regulation of body temperature, cyclic activities of the body, including sleeping and waking cycle. The nuclei of the hypothalamus are divided into magna and parvocellular depending on the size of their neuron pericardia. Neurons in some nuclei of the periventricular zone produce peptides into the blood circulation or, in some cases, into the cerebrospinal fluid. The process of the secretion of such bioactive substances by neurons is termed neurosecretion, and such neurons are called neurosecretory. They have a light nucleus with well-defined nucleolus and basophilic cytoplasm that contains a developed granular endoplasmic reticulum and Golgi complex. Parvocellular neurons in the nuclei of the periventricular zone produce releasing hormones, liberins and inhibiting hormones, statins, whose main purpose is to control the release of other hormones by the anterior lobe of the pituitary gland, adenohypophysis. Axons of neurosecretory parvocellular neurons form terminals on the primary capillary network, so-called neurovasal synapses, in the median eminence that belongs to circumventricular organs. Produced hormones are transported along the axons to these terminals and enter the blood circulation. Processes of tenocytes and on the capillaries of the median eminence, and they are able to regulate, close or open the excess of hypothalamic hormones into the vessels. The capillaries gather in the portal veins that form a secondary capillary network between the endocrinocytes of the adenohypophysis. Axons of magnocellular neurons in the supraoptic and paraventricular nuclei form the hypothalamo-hypophyseal tract, which ends in the neurohypophysis. Produced by these neurons, vasopressin, or antidiuretic hormone, and oxytocin are transported to the neurohypophysis and accumulate in axonal swellings near the capillaries. The largest axonal swellings are called herring bodies. Axonal swellings provide a local depot of secretory granules for release into the capillaries. The subthalamus is a small area of the diencephalon that lies between the thalamus and the midbrain. It contains several nerve tracts and the subthalamic nuclei, which are functionally associated with basal ganglia and are involved in controlling motor function. It will be told in the next video. Thank you for attention.